Good morning, everybody. So, welcome to the Dr. George Skoros special talk. So, this is the first time for him to visit in Thailand. So, uh, we are so lucky he have a time to give a special talk also, over here as well. So, before we start the talk, I would like to introduce a bit about Dr. George Skoros. So, well, Dr. George Goros, he is the professor in radiology and radiological science, radiation oncology and oncology at Division of Nuclear Medicine and Molecular Imaging, School of Medicine, Johns Hopkins University, Baltimore, USA. And he also served as the chairman of the Medical Internal Radiation Dose Committee of the Society of Nuclear Medicine and Molecular Imaging as well. So please welcome Dr. George Skiros.
Thank you very much. And thank you so much for the wonderful hosting that I've had on my visit. It's been a pleasure. First time in Thailand, and we've really enjoyed it. I'm in Bangkok with my wife, and I want to thank Professor Anshali and Dr. Kamwan for hosting me and having me stay at a wonderful place. So I'm very happy to be here to present. I'm going to, so usually in the US I have no pointers. So here in Bangkok I have a green pointer for important things and the red pointer for things that are less important. So I'll go ahead and start. Again, thank you very much. My talk is Precision Medicine and Radiopharmaceutical Therapy. I want to start by stepping back. So let's first think about cancer and cancer treatment and how patients are treated for cancer. So if the cancer is local, has not spread, then surgery to remove the tumor works much of the time and can be very effective, or radiotherapy if it's more difficult to do the surgery. So the combination can be very effective in treating most cancers. There are some exceptions: pancreatic cancer, brain cancer, some other cancers, ovarian. So there are many exceptions to this. But for many cancers, this works very well. The problem really happens for almost all cancers after the cancer after the cancer has spread or metastasized. Then when the cancer is spread throughout the body, surgery doesn't really work. Radiotherapy is difficult. Still, after many years, the main treatment is chemotherapy, or more recently, there's treatment, biologic therapy, which is, and I'll describe a little bit more what that is. So chemotherapy, everybody knows about chemotherapy, right? It's very toxic. It doesn't work all that well. It kills rapidly proliferating cells. Targeted biologic therapy is the idea that tumor cells depend on signals to behave like tumor cells. And if you have a drug that stops the signal, then the tumor cell can go back to being a normal cell. So it doesn't really kill cells, but it is targeted because it'll only affect cells that have the pathway that makes them behave like cells. And then immunotherapy is a new and exciting area with the checkpoint inhibitors, and this is going to become more and more interesting as we, in the future. So what I'm going to talk about is radiopharmaceutical therapy. And it has properties of chemotherapy and also of targeted biologic therapy. So radiopharmaceutical therapy kills cells like chemotherapy, but it does it in a targeted way, like biologic therapy. So, and it does it by using radiation. So we all understand radiation, and we know how that works. In a way, it has the best properties of both. I'm going to spend a little bit of time talking about chemotherapy and then a little bit of time talking about biologic therapy to kind of show you how these all are different. So chemotherapy is basically how many different drugs that kill rapidly proliferating cell can you put into a patient before the patient can't tolerate anymore. And there are different ways of killing cells, and I've just shown some of these here. The main feature of chemotherapy is that it has high toxicity. And in fact, the toxicity is so strong and so important that in the world's knowledge of Wikipedia in 2014, the page about the toxicity was translated into many different languages, including a language called Simple English. So this is really the key feature of chemotherapy. It kills all cells, but it's very toxic. Targeted therapy is a different approach. So it looks for pathways in cells that depend on maintaining their cancer type. And some examples are Gleevec and Iressa. So these were drugs, very successful, a lot of excitement. They had a big impact, but then the tumor cells found ways around the pathway. So in the end, even though there was a lot of promise and a lot of success, they haven't been as successful as everybody hoped. And just to show you why that may be and give you a little bit more background, this is sort of from the Hallmarks of Cancer paper by Weinberg and Hallahan. So when I talk about biologic therapy, right, this is an example of the signaling pathways. So let's say we're looking at cell proliferation. So the way that a cell knows to keep dividing and to keep proliferating is that it receives signals. Okay, so let's see, maybe the green one. So it receives signals from outside that tell it to grow or tell it to die. And then these signals go through a pathway, and it's like instructions. It tells the cell what it should do based on what it senses from the outside, or sometimes what happens on the inside. So let's see. So here's a DNA damage sensor. So if the DNA is damaged, the cell, the machinery of the cell sends signals to say there's some damage, we have to fix it, we have to repair it, and then you have a cascade of different things. This is really one of the ways that treatment is being implemented in cancer therapy. So one thing I want to say is that I'm a physicist, but you know, I think that as physicists, we need to think more and more about the biology. So I'm starting out with a lot of biology, and I think in general, we need to really be diverse in our understanding. So not to just stay in one area, but to think in a bigger picture and understand what's going on in other areas, because that's how you succeed. That's really what makes for a successful research approach. So anyway, so this looks complicated, but it's actually much more complicated. So if we focus on one pathway, somewhere here is AKT. Let's see, let me put my glasses on so I can find it. Okay, so let's say we focus on one little pathway here, is P10 AKT. If you just take this little pathway here, it's actually all of this. So very complicated. And biologic therapy works by interrupting one of these pathways, and that then changes how the cell behaves. But of course, over time, the cell can find a different pathway and get around that break. Okay, so that was chemotherapy and biologic therapy. Very quickly, I mean, this is a big topic, but I very quickly presented it. So now let's talk about radiopharmaceutical therapy. When people talk about, in nuclear medicine, radiopharmaceutical therapy, they talk about guided missiles and delivering radiation to tumor cells in a way that's very guided. But this is really, if you think from the physics, this is how it works, right? So let's say this is a patient and this is the body. You have some tumor cells. The first step is you administer the radiation, the radioactive molecule, the radioactive agent, intravenously, systemically, distributes throughout the body, and then it clears over time. So here's the clearance. And when the molecule encounters a receptor that sticks to a tumor cell, then the radiation stays on those molecules. So in radiopharmaceutical therapy, the key question for success is this one. Where else does the drug concentrate and for how long? So if you think of what is important for success of a treatment, in external beam, success depends on how well you can target the beam and how much you can control for the motion of the patient, and how much radiation you're delivering to normal tissue. For radiopharmaceutical therapy, the key question is really the pharmacokinetics. Where is the molecule going? How long is it staying there? What kind of radiation is it delivering? And it's delivering radiation, and we understand radiation very well, more or less. So we can calculate the doses, and we can project how much we need to deliver. Okay, so now let's talk about precision medicine. In 2015, President Obama said a sentence that included the word precision medicine, launching a new initiative to bring precision medicine, give it access to personal information to keep ourselves and our families healthier. So as soon as this was in the State of the Union address, as soon as the president says something like this in the US, a lot of things happen because every word in the State of the Union address has been studied very carefully to make sure that there's a policy behind it, that the government will do something about it. So the first thing that happened is that companies started talking about precision medicine. So here's a company right away. Once precision medicine is said, then the whole field starts saying, let's talk about precision medicine. And then the government basically explains what the policy is going to be. So precision medicine is to advance pharmacogenomics with the idea that the right drug to the right patient at the right dose. So dose is very important here because I do dosimetry. And if you think about dose, everything I've described about radiopharmaceutical therapy requires understanding dosimetry and dose. So let's talk some more about radiation dose in nuclear medicine. Everything that we think of in nuclear medicine about radiation dose is really for diagnostic imaging. So the reason we want to know about the radiation dose is to go from absorbed dose to biologic effect. And then the question is, once you have the dose, how can you convert it to a biologic effect? If you're interested in diagnostic imaging, bone scans or brain scans, then your question is the risk of cancer, not really anything else. And the data to go from this to this come from epidemiological data, so whole organ dose estimates. There are tables, the ICRP publishes tables where you can take the average organ dose and understand the risk of a diagnostic approach. For therapy, we're interested in efficacy and toxicity. Is there a clock here? How am I doing in time? Okay, so I'll go a little bit faster. So in therapy, we're interested in efficacy and toxicity, and the input for that is preclinical radiotherapy, external beam, preclinical and phase 1 studies, and we want to know not just the average, but the dose distribution. We need the radiobiology and efficacy and toxicity. Okay, so this is the dosimetry that has been used for diagnostic imaging, MER dosimetry. So if you want to know the dose from, let's say, the lung to the liver, you need the number of disintegrations in the lung, the energy released per disintegration, and the fraction from lung to liver, and then divide by the mass of target. So let's go through each one. First one we get like this. We collect images over the tissue, integrate over time, and then we get the residence time. The second one we just look up in a book. It's a property of the radionuclide. So let's say iodine 131. So this is total energy for photons, total energy for electrons, and total energy emitted. So that goes over here. This times this is the total energy coming out of an organ. But that's not the dose, right? So the dose is the fraction of that is absorbed in a target. So that comes from a Monte Carlo simulation. So photons travel a large distance, and the self-dose is less than 1 but greater than 0, the source to target. So let's say the fraction from the liver to a kidney is going to be, could be 0 and will be less than 1. But if you have electrons, short range, or alpha particles, cross organ is 0, but self-dose is 1. This is the MERD method for diagnostic imaging, not so much for therapy. Okay, and then we divide by the mass. So to make all this work, right, we have all of this, and then there are tables that you can use to do the calculation. So the tables are S values, but because we have geometry, we have the mass, and we have the radionuclide, you need a model of the geometry. And that's what is still being used. So this simple geometry doesn't have tumors. It doesn't allow for smaller distributions. So then the dose is just this equation here, the standard equation. So if we want therapy, then we need to do patient-specific dosimetry. And this is a software that I developed quite a while ago in '92. And then more recently, we added radiobiological modeling. So here's the radiobiology. So this is to go from absorbed dose to biologic effect. The biologically effective dose accounts for the difference in delivering radiation over 1 day versus delivering a total dose over 1 week. So if you deliver the dose over 1 day, the biological effect is greater. If you deliver it over 1 week, biological effect is lower. And the math is here. So this has radiobiological parameters and a repair constant for DNA damage repair. The equivalent uniform dose takes into account the idea that if half the tumor has 100 gray radiation, the other half has 0 gray radiation. The average is 50, which sounds like a lot, but the part that has 0 gray is going to come back, and the 50 really doesn't mean anything. The response is going to be influenced more by the part that's getting no radiation. So the equivalent uniform dose is not going to be 50, it'll be closer to 0 or maybe 1 or 2 gray to really reflect the idea that that the dose distribution is not very good. So, okay, so here's the package. We take SPECT or PET image information and CT or MRI. We do registration. We have activity at different times, density from the CT and composition from the CT, calculate some parameters. And for the quantification, the MERD committee has published pamphlet 23, which you can look up and gives details on how to do that. So here's an example. This is samarium-153 osteosarcoma patients. This is an image of osteosarcoma. It's an interesting problem from dosimetry because osteosarcoma is soft tissue and bone. So the dosimetry is a little bit complicated. So we did the 3DRD dosimetry, and this is showing the absorbed dose to the tumor for patients that progressed, progressive disease, and for patients that had stable disease. So a very clinical measurement, progressive versus stable disease. And you see that both the absorbed dose and the equivalent uniform dose, have a difference. These are all individual tumor or individual patient responses. So if the tumor dose was greater than about 18 or 20 gray, then the response was stable disease. If it was below that, then the patients progressed. So this is a clinical measurement, and it was significant. If we want a more physics measurement, then here, each point here is the change, the percent reduction in tumor volume from CT scans. So we see the same kind of thing now with a very physics, much more quantitative measurement. You see that at around 21 gray, these points, negative means the tumor grew. The tumors all grew if the dose was less than 21. If the dose was greater, then either they stayed stable or they shrunk a little bit. And the same thing for equivalent uniform dose. So, and statistically significant. Okay, so we know from radiotherapy that to treat osteosarcoma successfully, you need 69, 70 gray for a 3-year remission. So what I just showed was about, and because radiotherapy is delivered uniformly, that means that the EUD must be 69. So the EUD I showed was about 6 gray. So very far from being effective, just with samarium-153. All these patients did not do well. So how can we improve this? So we said, well, we know that samarium will deliver a high dose in very specific regions, but not high enough. And we know that external radiotherapy is very good at delivering high doses in a large region. So maybe we can combine radiotherapy with external beam radiotherapy. So this is kind of the example that we think about. Here's a patient with a lesion in the spinal column, and radiotherapy alone maybe won't deliver 70 gray because you have the spinal cord limiting how much you can deliver. But we know with samarium we can actually deliver a lot to the tumor, maybe not uniformly, but to sort of give a boost. So the idea is that if we combine the two, we have to adjust the dose from radiopharmaceutical therapy to make it into 2 gray per fraction, so that when we go to the medical physicist in radiation oncology, we give them a dose map or a DVH that's in the same, that is in the dose values that they think about. So this is the equation that we use, and it's the biological effective dose equation. So here's the dose from the radiopharmaceutical therapy, and then we convert it to some number of gray per fraction, and then there's a bunch of math to do that. So here's the G factor, the Lee-Catachide equation. So here's a dose volume histogram now for samarium in 2 gray equivalents. So we took the step to go from dose in nuclear medicine to dose the way that the external beam people think about it, depending on how they do their fractionation. This is the contribution from radiopharmaceutical therapy, not very good, 22 gray, but with about 7 gray to the spinal cord. So if we take that, we combine it with IMRT that delivers, a certain dose, and this is the IMRT plan, we can increase the external beam plan to deliver a treatment plan that stays inside the limit for spinal cord toxicity, but delivers more dose to the tumor than you would otherwise. And here's a picture for a different patient of how this works. So the bright red is the samarium, and everything else is the external beam. So this combination with the radiobiology makes it possible to treat more difficult cases than each one by itself. And the way to do that is you really need the radiobiology. You can't use whole organ dosimetry, you have to understand the dose distribution. Okay, so I'm now going to talk a little bit about alpha particles. Okay, not too bad. So, so this is a slide I always show. Alpha particles have a high LET compared to beta particles. A small number of tracks will kill a cell. The damage is irreparable. So here's what an alpha particle looks like. This is a bubble chamber image. These are electrons. Here's a cartoon showing beta particle has a large length, deposits little energy. You need many tracks through a cell to kill a cell. An alpha particle has a short range, very potent, and can be very effective at killing a cell. 2 or 3 tracks can kill a cell. So we've used bismuth-213, 45-minute half-life, comes from a generator, actinium-225 generator. The radiochemistry has been worked out. And this has been used in a lot of preclinical studies and also some human studies against leukemia at Sloan Kettering. Actinium Pharmaceuticals is running a trial with the parent of this. So it's been looked at in Substance P. There was some famous work in Basel against glioma. So this is an exciting antibody, an exciting alpha emitter, but the problem is very short half-life. Makes it difficult to use. The radiochemistry has to be very fast, and the patient has to be ready. So instead, people are using actinium, 10-day half-life. It's the parent of bismuth, has 3 alpha-emitting daughters. You can get it from the decay of uranium-233 or thorium-229. And now, more recently, some cyclotron methods are being studied to try and increase the supply. And the chemistry is well developed. So this one now is in multi-center phase 1, 2 trials against the leukemia in older patients with acute myeloid leukemia. So in external beam radiotherapy, protons have become popular. So the reason is that protons, and like all energy, particles, have a high LET. So this is just showing you actinium-225 has 4 daughters. So here's actinium, and then francium, astatine, and bismuth. So each one, as it travels through tissues, has a Bragg peak. And then, so the energy deposition, the LET, is much, much higher, even at the beginning compared to electrons. So here's electrons, and here are the curves for energy deposition density as the particle travels through tissue. And this was a Monte Carlo calculation done many years ago by Dudley Goodhead that explains the idea that electrons, you need a lot of electrons. They're uniformly deposited to deliver 1 gray. But with alpha particles, through the nucleus, a small number of tracks, 4 tracks, can deliver the same dose. So that was Monte Carlo simulation. Now, with molecular biology, we can image this. So these are images of gamma H2AX. These are cell nuclei exposed to different, here it's external beam, here it's alpha particles. And if you look at, let's say, this group of cells, you see these nice straight lines coming from alpha particles. So this is, the yellow is all of the molecules that come together to repair DNA double-strand break damage. So it's not the track. We're not looking at the track. We're looking at the cell response to the damage, the molecular machinery that repairs the damage. So there are antibodies that you can label with fluorescent markers to look at this, and this is an established technique. This is not from my lab, but it's something that is relatively well known. So for alphas, you see this straight tracks, a lot of damage. For external beam, much more distributed damage. Okay, so the pattern of damage changes. So for, let's say you have 10 gray from alpha particles versus 10 gray from beta particles, the biologic effect is much greater from the alphas because of the way the damage is delivered. Mostly DNA double-strand breaks, and less so for the other, for photons or electrons. And the way that's expressed is in terms of a relative biological effectiveness. So to get this, you need a biological effect, you need a reference radiation, and the test radiation. So the test here would be alpha particles. So alphas are 3 to 7 times more effective at killing per unit absorbed dose than photons or beta particles. So this measurement, right? So what does this measurement depend on? It depends on the biological endpoint, X, the reference radiation, but it also depends on the dosimetry method. So if your dosimetry method is different for alphas versus betas, or if you're not doing the dosimetry correctly, then the factor will change. And it also depends very much on the biology. So I want to show you I talked about molecular mechanisms and how the cell senses damage and then causes repair. So these are two pathways that respond to DNA double-strand break damage. So this one is when the damage is very big and there's no template, so non-homologous end joining. This is homologous recombination, two different pathways. But if you go with molecular biology and you stop the pathway by inhibiting these different molecules, then the repair of the DNA double-strand break damage stops. Not completely, but it stops quite a bit. So what that means is that if you deliver alpha radiation with these repair inhibitors, you can kill cells much more efficiently. And here's, again, similar kind of thing showing 1 hour, 24 hours. Most of the signal is gone because of repair, but if you have a DNA PK inhibitor, then there's still quite a bit of damage. The important thing about this is people think of RBE as a physics thing, right? It depends on alphas versus protons versus electrons versus photons. And they think of it as a single number. But the biology makes a big difference. That's why I say that the physics community has to think more and more about biology. So if you have a way of biologically reducing DNA double-strand break repair by either a drug or by changing the pathway. So here's BRCA1. This is an important pathway for breast cancer. It has an effect on DNA double-strand break repair. You can go from about 3 to 5 RBE all the way up to 16, 15, 16. So this is not increasing radiosensitivity overall, right? Because RBE, by definition, the sensitivity to low LET damage has to stay the same for the RBE to go up. If both of them go up, then the RBE stays the same. So this is sensitizing the cell specifically to DNA double-strand breaks, alpha particle damage, which means that you have the possibility now of delivering in a very targeted way an alpha particle emitter, combining it with a drug that inhibits double-strand break damage and not impacting all the other cells that experience other kind of damage. So so there are many drugs now in the clinic that are looking at that have this effect. They inhibit specifically double-strand breaks. So PARP inhibitors are an example. There are many drugs in clinical trials that are using this. So it's an interesting approach, and it's something that I think requires a lot of research, and it really shows that in nuclear medicine, therapy has a lot of potential. I think there's a lot of exciting things that can be done because the treatment approach is so flexible. But to make it all work, You need physics, you need biology, you need chemistry. You really need a multidisciplinary approach. So it's really a very exciting area to think about for the future. Okay, so I said I would talk about radium. I'm going to say just a few things. Here's radium-223, 11.4 day half-life, 3 alpha particle emitting daughters. The first one is radon-219, which is a gas. It works because its radium chloride behaves like calcium. It was first made by Algeta, but now it's sold by Bayer as Zofigo, and there have been a lot of preclinical studies. Right now, when I did the slide, there was a multi-center trial. The trial is finished. So now it's an FDA-approved drug for patients with castrate-resistant prostate cancer. And it works well, but as the patients get more and more docetaxel, the toxicity goes up. But the first studies had very low toxicity. And this is one of the early reports from ASCO describing alpha-radin, is what it was called at the time, targeted approach for treatment of bone metastases from castrate-resistant prostate cancer patients, calculated alpha particle dosimetry compared to a favorable clinical safety profile. So the reason I show this is because the dosimetry for alpha particles is not easy, it's complicated. And if you're a physicist interested in something challenging and new, then dosimetry for alpha particle emitters is something that I think would be very promising. So let's look at what this means, why they had this title. So in this trial, less than 1% of about 300 patients had grade 4 toxicity. 2 to 4% had grade 3 toxicity. This is better than radiolabeled antibodies. It's better than almost all radiopharmaceutical therapy, very low toxicity. So we calculated the dose in the conventional way. The absorbed dose to the red marrow was calculated as 1 gray, and then you put an RB of 5. So 5 gray with the RB adjustment. So 5 gray to the marrow would predict much more toxicity than we see here. And we did the calculation assuming the way I showed, assuming sort of a MERD whole organ kind of calculation, basically assuming that all of the activity is inside the marrow cavity, and then we say we say that all the activity is on the bone surface, and then calculate the fraction of energy emitted from the bone surface that's absorbed in the marrow cavity, divide by the marrow. So this is a calculation that is very simple. You can do it, but it actually does not predict the biology, because the biology in patients was much less toxicity than 5 gray. And we know that 5 gray is too high because we did many years ago a study for iodine-131 showing that if you have a red marrow dose of more than 3 gray, most patients have grade 3 or 4 toxicity. Almost all the patients have grade 3 or 4 toxicity. So if you deliver 5 gray, then everybody should have very high toxicity. So we didn't see that. So let's try and understand why we didn't see that. And once you think about it, the answer is quite simple. So here's a model of the bone. This is the bone. Here are some tumor cells. And this is the trabecular marrow cavity. So you could have the radionuclide distributed in the bone cavity on the surface or only on tumor cells. So radium behaves like this. It's only on the surface, and the range of the alpha particles is short. So even though it's in the marrow, it only irradiates a small portion of the marrow cavity, not the whole cavity. So all of this marrow is really not irradiated, so it can recover. And in fact, if you do the calculation, you can have a dose that's very high, but because of the geometry, and this is, once you think about it, this is obvious almost, right? Because of the geometry, you can have an average dose that's very high, but only 40% of the cells will get 2 gray. Most of them in this region are not going to experience radiation. So this is important in explaining the difference between a simple calculation and the biological effect. The other important thing, of course, is if you do this many times, the cells that you kill here are going to be replaced by cells that are normal, that were not irradiated. And if you keep doing it, then maybe you will have serious radiation. And in fact, now that the combination, and this is approved, there are examples where some patients experience serious toxicity. So it depends on how good your marrow is to begin with. Okay. So I'm near the end of the first talk here. So conclusions, radiopharmaceutical therapy provides targeted delivery of radiation. We understand how radiation works very well. It's not complicated like targeted biology, targeted biologic therapy. Depends very little on cancer cell pathways. It kills cells rather than preventing them from being tumor cells. If you use an alpha emitter, then you don't have as much possibility of resistance. And with imaging, you can do the kind of dosimetry that allows you to treat each patient individually. And then use that to understand the dose to tumors and to normal organs and guide escalation protocols and plan treatment. And in the next talk, I'll show this a little bit more. So radiopharmaceutical therapy may be a better choice for combination treatment with targeted therapy than chemotherapy. And radiopharmaceutical therapy in combination with radiotherapy is feasible, and right now we're doing it at Hopkins in clinical trials. So if you want to know more about alpha particle dosimetry, then the MERT committee recently published this book. It's available for purchase at the Society website. And if somebody's really interested, I have a PDF. If you send me an email, I can send you a free copy. But don't tell anybody. Okay, so this is the group in the lab. These are the collaborators. Kitty Watt worked in my lab for about a year on a different topic, pediatric dosimetry, FDG pharmacokinetic modeling. And we get actinium from Germany. The NIH, DOD, and DOE helped pay the bills for all the work I showed. That's it. Thank you. Okay. So the next one is shorter. It'll go faster, yes. Okay. Okay, so the next one is going to talk about thyroid dosimetry. So one of the questions, right, in treating patients with thyroid cancer is how much iodine-131 do you give? Fixed dose, a dose, not dose, but administered activity that will kill tumor cells, or one that will avoid toxicity. So there are, let's see. Right, these three basic approaches. Benoit-Leeper was a method developed to avoid toxicity that requires blood dosimetry and whole body retention. I'm going to talk about this. I'll talk about a method that we've used to investigate at the Hopkins. So let's look at each one. So if we want tumor absorbed dose, so the first one is easy, right? You just pick a number that from clinical experience. And sometimes it's very good, it works fine, but it doesn't really take into account the tumor and the kinetics and how each patient is different. The second approach says, well, I want to deliver what I think I need to kill a tumor, and if I know that in this patient I can't do that because the clearance is too rapid or the tumor doesn't take up enough, I will treat the patient a different way, maybe not radioiodine, maybe some other way. The last method is based on the idea that I know that if I treat a patient with a little bit of not enough dose, they're going to come back, I'll have to treat again, and the more I treat, the less uptake my tumor will get. So it's important to treat as much as possible the first time so that you can treat the tumor as much as you can, but without having toxicity to the patient. So the Benoit-Leeper method basically is, let's treat as much as we can the first time, but not so much that we have a patient that's very sick and has toxicity. Okay? So the first method showed that you need about 300 gray to ablate the thyroid and about 80 gray for metastases. And if you have less than 3 gray, you should use a different method. So the advantage is you don't treat patients for no reason, no overdosing, and you have a good reason to use a different treatment. And then you can also collect data on tumor dose response. The disadvantages are you need tracer administration, you have logistics, you have to image the patient, they have to come back, and maybe it's more expensive. The Benoit-Leeper method is based on the idea that if you keep treating patients with subtherapeutic therapy, you can lose radioiodine avidity. So the idea is find the largest administered activity that will be safe but optimally therapeutic, and it was formulated, it was published quite a while ago on clinical experience collected at Sloan Kettering. So that method says it's safe if the patient gets no more than 2 gray to the blood, and the blood is really being used to represent the marrow, and if the whole body retention at 48 hours is no more than 4.4 gigabecquerel. But if the patient has disease in the lungs, then you worry about lung fibrosis, and then you reduce the whole body retention from 4.4 to about 3 gigabecquerel. So where did all this come from? And I think this is really interesting because, you know, when we practice, we do physics, we think about certain rules that we just accept and say, okay, this is what the rule is. But if you look at the details, where did these rules come from? So these limits came from 15 patients, 11 female, 4 male, from this one publication. And here's some of the data. Okay, so this was a publication in 1957, and so some of these patients had lung disease, and 2 of them, let's see if, so 2 of them, I'm going to stand here, look a little bit better. They had a high dose, and in fact, high level of toxicity. So you can see that OF, let's see some of the, diffuse pulmonary metastases, and these had a grade 3 toxicity, and some of them actually died. So this is really the data that was used. A small number of patients published a long time ago with methods that are no longer relevant. So the idea is that sometimes it's good to think back and maybe re-examine, our assumptions about how we treat patients with nuclear medicine. Right, so here's, let's see, so some of these patients were given a single dose and they had grade 4 pulmonary function loss. Some were treated multiple doses, and just showing this is the, right, so this is the iodine 131. At the time in 1957, imaging was not available. So they could only measure whole body probe measurements. And this was the total amount in the body after 48 hours. And they found that if you keep going up, and this is in millicurie, if you keep going up, you have the risk that some of the patients are going to have severe lung fibrosis. Okay, so now I'm going to discuss more modern methods. So we've developed this 3D dosimetry package. You saw this slide already. And I've showed this already, the radiobiology I've explained. you saw this before. So here's a clinical case, a very special clinical case. This was a pediatric patient, a young girl with metastatic differentiated papillary thyroid cancer with a lot of lung involvement and also some lesions in the brain. When the patient came to Hopkins, she had already been treated with radioiodine. So she had iodine in her lungs. So we couldn't use imaging to understand to get a dose prescription. So instead we used I-124 with PET-CT to do a dosimetry assessment. We wanted to make sure that the amount of activity we give was not too much, to lead to lung fibrosis. In a pediatric patient, this is very important. So we used I-124, and we gave a tracer dose of I-124, and then collected 5 PET-CT scans. Here are some of the details. We used 3DRD and did all the stuff I showed before, with the idea that we would calculate from this how much activity we can give for therapy to not give more than 27 gray to normal lung. And we compared the method we used with other conventional methods. Olinda, I'll talk about Olinda, I won't talk about Benoit Leeper. So here are the images from the PET-CT. You can see a lot of lung disease, lesion in the brain. This is, let's see if I can do this. So I skipped a lot, and I'm just showing you the, so this is the dose map. So the intensity here corresponds to the absorbed dose in the lungs. And then we have a dose volume histogram showing the results. So we calculated about 5 gigabecquerels so that we don't exceed 25 to 27 gray to the lung. And this dose would give 325 gray to the lesion in the brain. So we thought that would be a good approach. The lung tumor dose turned out to be about 37 gray. Equivalent uniform dose, much less. But this was one approach that we used to try and treat this patient. So we compared it. And this is interesting for us because we did the calculation, not retrospectively, but in time to treat the patient. So using a Monte Carlo relatively complicated dosimetry, we were able to do a calculation in time to have an impact on patient care. And since this is not FDA approved, we just talked to the physician and explained how we did the method, and then let the physician decide how much to administer to the patient. So in fact, the patient did get 5.1 gigabecquerel. And the patient did well. This is one case. It doesn't really prove anything, but the patient did well and everybody was happy, and now the 11-year-old has graduated high school and is living a happy life. So we compared it with Olinda, and this is really the important part of the presentation. Because we have a method, we need to explain how it works. So for the thyroid calculation, we had 2 contributions, right? One from the lungs and one from the remainder body. So if you remember, I showed you the curve. We calculated we had all the time points, we integrated. We had one term for the total dose to the lungs coming from lung to lung, and one coming from the rest of the body to the lung. But this was for a child, so we had to take the phantoms that are used in Olinda and fit them to different whole body masses, and then we knew that our patient had this mass, so we calculated the absorbed dose, and then for different phantoms, and then picked this as the phantom, and then used this to say, if we don't want to deliver 27 gray, then Olinda predicts about 3 gigabecquerel. So Olinda gave about 3, 3DRD gave about 5, so there's a difference. So we went back to understand the difference, and this is part of validating. If you can explain the difference, then that really gives you some knowledge. So if you remember the images, right, the CT, this was a patient with lung disease. So the phantoms in Olinda are for normal patients. We adjusted for whole body mass, but the lungs in this patient were heavier because they had tumors in them, not all air. So if you take the same approach, but now instead of whole body, adjust for the lung mass. So again, kind of simple. You have an answer that comes much closer to what you calculate with 3DRD. the point is that a patient-specific method using CT already takes into account the density, the difference in the lung mass, because you're using CT and you're using the Hounsfield unit to adjust the calculation. But when you take a standard approach, you don't get that. Unless you recognize that, unless you recognize that the difference in lung mass is important for these two different patients, and then make the adjustment. So this is an example where patient-specific calculation can give a very different result for a reason that you could never predict. I mean, it would be very difficult to predict. Okay, so we showed that 3DRD was feasible. We did it in real time by getting the image as soon as the patient finished, running the Monte Carlo, doing the calculations in real time. We got a higher value than by an S-value-based method. We looked at why the difference was, and we explained it as the difference in the lung mass. So what I showed you was dose to normal lung and dose to tumor. So the problem there is, and this was done by a voxel to voxel calculation. So we need a lot more research, because in a single voxel on a PET scan, you could have both tumor and normal tissue. So the resolution is not enough to distinguish between, is this voxel tumor only or normal tissue only? So we're working on this method. The way we did it here was combination of CT density and activity. So this is something that's an area for more research. And here are some results. We continue doing this. We've collaborated with a group at Washington Hospital Center. So here are lesions, and we're doing these dose volume histograms. We're getting different dose estimates, and we're calculating EUD. Another example. And then we can do this for normal tissue, lungs, kidneys, liver, spleen. So this summarizes the experience with Washington Hospital Thyroid Center, where they do a lot of I-124 PET imaging. And what I want to show here is, if you look at, let's say, tumor 3 and tumor 11, this is the absorbed dose, 110 gray. This is equivalent uniform dose. So if you look at these numbers, you would say, well, this is 110 gray, so probably tumor 3 will respond better than tumor 11, because tumor 11 has less dose than tumor 3. But if you look at this column, the equivalent uniform dose, the dose distribution, is better for tumor 11 than it is for tumor 3. So less of this tumor is getting a low dose, even though the average may be higher for this. So the idea here is that with radiobiology, you can better predict the likely outcome of a patient. So again, this is, not definitive, doesn't prove anything. It just says that if we do it this way and collect more data, maybe we can start better predicting, how patients will respond to treatment. So like I said, we need to correct for small tumors, because we're doing voxelized calculations, and there are many difficulties with that. In some cases, the tumor is not visible on CT. We need more patients, and to really prove this, we need to follow up response versus dose, so tumor volume and FDG changes. And other things we're looking at are salivary gland toxicities, and how to make this all work much better and faster, so you don't have to have a big physics group all spending a lot of time doing these calculations. So this is the group that worked on this. Paul Adenson was the endocrinologist that was involved in this study. Doug Van Nostrand is the physician in the Washington Hospital Center that sent us the I-124 PET data that helped us do all this work. Thank you.